Hello, everyone, and thank you for joining us today. I am pleased to welcome you to the TriLink Biotechnologies mRNA Basics webinar series. Today's webinar topic is Strategies to Minimize the Innate Immune Response. Our presenter today is Corey Smith, Senior Technical Sales Specialist at TriLink. Corey has worked with mRNA technologies for several years, both at the bench and as a resource for our clients on their mRNA therapeutics development path. He's very familiar with strategies to minimize the innate immune response when designing mRNA therapeutics, and will be able to take your questions at the end of today's presentation. With that, I will hand the presentation over to Corey.
Thank you, Dina. So as Dina mentioned, I'm Corey Smith, a Senior Technical Sales Specialist here at TriLink. I'd like to welcome everyone to our first iteration of the new mRNA Basics webinar series. Today, I'll be giving a presentation and taking technical questions at the end. Please use the questions panel to submit questions throughout the presentation, and we will get to as many as possible. Also, here you can find the mailing address for the list to sign up for new information regarding our future topics for the webinar series. So for today's agenda, we'll be discussing how a cell's innate immunity can affect your in vitro transcribed mRNA. We'll be going over strategies to avoid this immune response to help increase expression. Some strategies include the use of chemical modifications, mRNA capping strategies, and different purification strategies. First, let's get into a little about who TriLink is. TriLink offers all of your nucleic acid needs, both catalog and custom. We offer oligos, NTPs, mRNA, and we have now officially launched our plasmid services department. We aid in the research of our clients by providing support for your manufacturing needs. We have GMP facilities and experience manufacturing these products for clinical trials. Here's our beautiful new facility that we moved into at the beginning of 2020. It was specifically designed for custom manufacturing and has ISO Class 7 and ISO Class 8 GMP suites with Class 5 fill finish hoods. We have 15 total GMP suites for different manufacturing projects. We have over 118,000 square feet with adjacent expansion space available. Here is a full list of our offerings with a bit of detail for each specific product. For this specific webinar, the Innate Immunity Response webinar, I want to point out that we manufacture NTPs with modifications. We also have a proprietary capping reagent, CleanCap, that co-transcriptionally creates a naturally occurring Cap1 structure. We also have the ability to use all of these reagents in our custom mRNA builds, both at the research grade level and the GMP level. Okay, well, let's get right into this. How can mRNA impact the innate immunity of your cells? So cells have several receptors, such as toll-like receptors 3, 7, and 8, to differentiate fully functioning mRNA from improperly formed mRNA. Double-stranded mRNA, improperly capped RNA, and DNA ends are not recognized as self by the cell's immune system. These are not fully formed mRNA and can activate the cell's immune system, leading to the degradation of the mRNA, lowering the levels of expression of your target protein. So it's very important to synthesize your mRNA correctly. In vitro transcribed mRNA that's not synthesized correctly can exhibit some of these features, which can trigger the immune defenses, leading to poor mRNA performance. However, today we'll go over strategies to optimize and design your mRNA to mitigate these effects. So the first that we'll discuss is chemical modifications. Chemical modifications are one of the easiest ways to aid in the immune response mitigation, and the #1 way is through base modifications. Modified NTPs can reduce the binding to innate immune sensors during in vitro transcription. There is evidence that they can reduce toxicity, leading to prolonged expression in cultured cells. And not only has this effect been seen in vitro, but it does translate to some in vivo applications as well, which we will touch on later in this presentation. The modified NTPs can lead to sustained expression of your target protein, and some of the most popular of these are modified uridines, including pseudouridine, N1-methyl pseudouridine, and 5-methoxyuridine. I think this is a great slide. It depicts GFP fluorescence across multiple cell lines. In each case, the GFP has been synthesized with a different modified nucleotide. You can see that the modified uridines, pseudo-U, N1-methyl pseudo-U, and 5-methoxy-U all perform quite well, most notably against their wild-type counterparts, as well as other modified nucleotides. Again, this shows evidence that modified uridines can play a significant role in mitigating the immune activation often seen when using wild-type uridine. And you can see just based on, graphically here, in just about every case, the N1-methyl pseudo-U performs significantly better than the rest. And in some, 5-methoxy-U and pseudo-U are up to par with that. But in just about every case, they are significantly outperforming their wild-type counterparts. So with this evidence in mind, that innate immune activity can be linked to uridine content, another way to limit the immune response is to codon optimize your sequence to deplete for uridine content. This would be called U depletion, and maybe some people have heard of it, but it's a way to really take a look at your sequence ahead of time, ahead of the mRNA transcription process, and determine if it's going to be optimal for your final system. The experiment here shows the difference in protein activity between standard luciferase sequence with modifications and U-depleted Luc sequence, also with modifications. So kind of the secondary measure here is we wanted to measure both a standard sequence and a U-depleted sequence, and the modifications thrown in were to see if that would actually affect it as well. In each case, U-depletion of the F-Luc sequence shows higher levels of luciferase activity. When paired with the base modification, specifically modified uridine, you can see that the expression increases even more. So this is really, really helpful evidence to show that not only is a base modification helpful for your use, but also you depleting your sequence can really help with the expression of your protein. In pretty much every case here, the U-depleted sequence are expressing almost 10x of what we would see with the wild type or with the standard sequence. So as a quick summary, here are kind of my main takeaways to consider when thinking about using modified bases for your mRNA production. One, chemical modifications are a simple way to design your mRNA to help to mitigate innate immune activity. The second is uridine content has shown to cause immune activation, which you saw through those two previous experiments. Codon optimizing to U-deplete your sequence is also a great way to aid in immune suppression. Lastly, I think it's important to note that different cell lines or systems may require different optimization techniques to aid in the immune response. Modifications to uridine and U-depleting are a great place to start, but depending on your final application, it may require a few head-to-head comparisons to determine what works best in your specific system. There's really no best way to figure this out besides really getting in there and doing it. But I think that your modifications uridine and U-depletion are a really great place to start on the front end of your mRNA development. Okay, we can move on to a different approach that can certainly aid in minimizing the immune response of your mRNA, and that is the implementation of different mRNA capping strategies. So there are two widely used cap structures for the in vitro transcribed mRNA. Those are Cap0 and Cap1. On this side, you can see the structure of a Cap0 mRNA. The Cap0 structure most notably has an OH group on the first sugar. It's important to note that this is not the structure of a fully formed mRNA, and it is not found in higher eukaryotic mRNAs. This is notable because Cap0 mRNAs will not be recognized as self by the cell's immune system, which can trigger an immune response leading to the degradation of the mRNA. Conversely, one simple change, the 2'-O-methyl group here, shows the structure of a Cap1 mRNA. This 2'-O-methyl group takes the place of the OH group in the Cap0 structure and is actually the proper and natural cap structure of eukaryotic mRNAs. This will be recognized as self by the cell's immune system and alone will not trigger an immune response. It's very important to note this, that there are obviously other factors that can trigger an immune response, but the cap structure and matching it to what is naturally occurring in a eukaryotic cell system is a really great way to not necessarily fool the cell, but make the cell understand that this mRNA is self. So again, the key takeaway here is that Cap0 will be recognized as foreign or improperly formed. IFITs can recognize this and sequester the mRNA from translational machinery. This leads to a significant decrease in protein expression and can lead to degradation of your mRNA. Cap1 mRNAs do not show this same issue, and in fact, the methylation can reduce binding to pattern recognition receptors. This again allows the cell to recognize your mRNA as self and can extend expression by not degrading the mRNA early on. So here are some of the historical capping strategies employed by RNA scientists over the years. Starting with enzymatic capping and moving on to co-transcriptional capping methods such as MCap, ARCA, and now CleanCap. So for enzymatic capping, pretty much the functionality and the way this works is through two post-transcriptional enzymatic reactions. And I want to stress that this is post-transcriptional. This is after you have your mRNA out of the IVT reaction. It's possible to form a Cap1 structure. This is done through a vaccinia, usually using a vaccinia capping enzyme followed by a 2'-methyltransferase enzyme. These enzymes needed for the reaction can be prohibitively expensive, leading to issues with scaling up the production. Additionally, the added enzymatic reactions lead to more handling of the mRNA, which is normally a quite delicate and often prone to degradation. This post-transcriptional handling can lead to a lower purity product. It's important to note in most cases at TriLink and in general, we really try to minimize the post-transcriptional handling of your mRNA. We want to make sure that what we get out of the transcription reaction is going to be really close to the purity we want. Maybe we need to remove a few smaller truncated products or some of the enzymes, but we want the mRNA to be, as pure as possible. So when moving into the realm of co-transcriptional cap analogs, the first generation to come was Cap and MCap. There were quite a few issues with Cap and MCap, whether they were binding to the mRNA in reverse orientation. There were issues with yield and capping efficiency. So there was a new kind of generation that came about, and that was the second generation, which was ARCA capping or anti-reverse cap analog, which I'm sure many people are familiar with if you've been working with mRNA. It's a pretty common cap analog out there. It's been around for quite some time, and it's been quite successful for quite some time. Because these are utilized co-transcriptionally, there's no need for these post-IVT enzymatic reactions. But there are some drawbacks. Due to the transcriptional conditions required for their use, the capping is inefficient and the yields are often significantly lower. Additionally, and most importantly, these type of cap analogs, ARCA and MCap, only produce a Cap0 product, which, as we have discussed, is not naturally occurring. The latest method of co-transcriptional capping is CleanCap. This is the third generation. This is what we like to call the third generation. CleanCap was developed by TriLink, and it was designed to function under the standard conditions normally required for IVT reactions. Because of this, CleanCap is able to maximize yield as well as capping efficiency and can produce a naturally occurring Cap1 product. That is very important because we're able to, A, cut out the post-transcriptional enzymatic reactions, but also maintain a Cap1 product with a co-transcriptional cap analog. So here's a little bit more about ARCA and some of the experimental conditions required for IVT capping. So ARCA preferentially binds to a G start like uncapped RNA, but this causes competition between the ARCA cap itself and some of the NTPs, specifically GTP, and it requires the starvation of the GTP from the IVT reaction. This significantly lowers yields and capping efficiency in ARCA capped products. ARCA tends to cap around 60 to 80% of the mRNA and usually yields around 2 mg per mil, 2 mg per mil of IVT reaction. So CleanCap here, on the other hand, we don't see this problem. The recognition site is AG rather than G and does not see the same competition as seen with ARCA capping. There's no need to starve the reaction of GTP, allowing for transcripts utilizing CleanCap to see yields 2.5 to 3 times higher than those using ARCA. Additionally, CleanCap tends to cap with Cap1 structure around 95 to 99% of the mRNA. So finally, in summation, here's the head-to-head comparison of CleanCap to legacy cap analogs, and in comparison to enzymatic capping as well. CleanCap is able to provide reduced immunogenicity when utilizing a Cap1 structure. It also yields about 3 times the amount of capped mRNA when compared to ARCA, as well as showing significantly higher levels of capping efficiency. Due to this, we're able to decrease the cost to achieve the same amount of functional mRNA. Compared to enzymatic capping, this is a highly efficient single pot reaction, which allows for streamlining of the manufacturing process, making scale up to larger batch sizes significantly easier, and in comparison to enzymatic capping, significantly cheaper. You don't have to deal with those expensive enzymes. You don't have to deal with the post-transcriptional handling. You will get a more pure product, and it's going to be cheaper in the long run than using all of those enzymes. So, as we previously mentioned, we're going to get into some in vivo application data here, because seeing in vitro data is great and it works, but most people's end goal is to get into in vivo applications or get into the clinic. So it's important to go over some data here as well. So in this project, we compared LNP, or lipid nanopackaged luciferase mRNA, with both Cap1 and Cap0 structure, as well as different purification and modification strategies. The readout was we measured luciferase levels in mouse livers when measured at 6 and 24 hours post-transcription, or post-IV injection. As you can see, the cap strategy and modifications play a significant role in luciferase expression. Samples performed best that had the 5-methoxy-U modification and a Cap1 structure, which is pretty in line with what we expected based on some of the previous data that we were showing. We were able to see strong fluorescent signal in this category even 24 hours post-transcription. The next that performed best was the HPLC purified using wild-type bases and a Cap1 structure. But the main takeaway here is that the chemical modifications used in the IVT reaction, being a Cap1 structure and a 5-methoxy-U, significantly aided in protein expression, even more so than the mRNAs that were purified via HPLC. And if you're looking, you can see that the Cap0 structure here in both cases, after 24 hours, was just about the same as the PBS control, while this Cap1 structure, the 5-methoxy-U modified version, is still really expressing strong levels, almost the same as the 6-hour here. So lastly, and I'm touching on this last because this is post-transcriptional, pretty much everything we had talked about before had been either pre-transcriptional design or during the transcription reaction. I'll discuss some purification strategies here that can help reduce immunogenicity or immune activity. So purification of mRNA is really meant to remove salts, NTPs, cap analogs, proteins, truncated or fragmented mRNA products, residual DNA, or double-stranded RNA. Some of the more traditional small-scale approaches can include silica membrane purification, which is basically a typical spin column based purification with a membrane that catches your RNA and filters through the impurities that you don't want. There's other types of purifications as well, such as precipitations. Many people are probably familiar with lithium chloride precipitation, but these are really good for small-scale projects. But it still lends to the idea that one of the most difficult aspects of scaling up your production is optimizing your purification strategy. So as I mentioned, silica membrane purification is not necessarily scalable. It's very labor intensive, and often in commercial settings, chromatography is a more widely used process. TriLink has developed two forms of chromatographic purification methods: LC isolation and reverse phase HPLC. LC isolation can remove residual protein, DNA, and NTPs from the system, and it's often done in tandem with a form of TFF or dye filtration. Reverse phase HPLC can decrease dsRNA, which is double-stranded RNA we discussed earlier on that can cause an immune response. So it can decrease dsRNA and enrich for full-length mRNA. But there are a few drawbacks here. Because reverse phase HPLC is an extremely precise method of purification, there does incur quite a large yield hit. This requires you to increase the starting scale of production and can really add to the cost of the production overall. Additionally, longer RNA sequences can become more difficult to purify with this method, as there is a column involved that's heated to a temperature. Denaturing of longer constructs can be an issue here. This is a slot blot readout. We've discussed that dsRNA can trigger immune responses. So slot blot is one of the main readouts that we use at TriLink for dsRNA detection. On this, the darker dsRNA standards are matched against samples to determine the qualitative amount of dsRNA concentration. The darker samples are showing higher levels of dsRNA, and you can see that reverse phase HPLC does indeed aid in the removal of dsRNA. The HPLC purified samples are performing pretty similarly when compared to the dsRNA standards, and even when using a modification such as pseudo-U, you're still seeing pretty high levels of dsRNA, even compared to the wild type or compared against the HPLC. So here we wanted to test a comparison of HPLC and non-HPLC purified Rhinella luciferase, and we measured the activity of each sample. Additionally, to add another wrinkle into this, we compared wild type 5-methoxy-U and pseudo-U modified samples. The wild type samples clearly tend to favor HPLC purified lots. The pseudo-U modified samples also seem to show favorability to the HPLC purification, with both iterations having an increased activity level when compared to their wild type counterparts. Interestingly, though, the 5-methoxy-U modified samples actually show favorability to the non-HPLC purified samples. This lends us to believe that often with the use of 5-methoxy-U, we can see increased expression levels without the use of HPLC purification. So if this is implemented properly on the front end, this can work to minimize the use of reverse phase HPLC, allowing mRNA designers to maximize yield and expression. So all in all here, purification strategies can be a great way to post-transcriptionally aid in minimizing innate immune activity. LC isolation may produce sufficient quality material for your needs, especially if you're U depleting your sequence and using 5-methoxy-U substituted mRNA. But if dsRNA, double-stranded RNA, is a major concern for your application, reverse phase HPLC is always a possibility. It works quite well for this purpose, but you will incur a yield loss with this production. Most importantly, you should make sure to account for these purification techniques in your initial project design, because you will need to figure out your scale on the front end, figure out what you're going to need, basically what you need for your readout, and so on. And pretty much just to wrap everything up here, here's some of my key takeaways. So number one, control of the innate immune activation is an important factor to take into account when designing an mRNA. This can lead to higher levels of protein expression and an overall more successful mRNA candidate. Chemical modifications, including U depletion and the use of modified bases, can help control this effect. These are both ways to easily optimize your mRNA production before and during the IVT process, eliminating post-transcriptional handling and purification. Number three, choosing the proper cap structure of your mRNA can significantly improve yields, capping efficiency, and can aid in protein expression. There is another modification that can be made during the IVT reaction to maximize your results. Purification can be a great post-transcriptional method to mitigate innate immune activation. And when necessary, reverse phase HPLC can be a great method to purify your mRNA. But it's important to note that for certain applications, some modifications such as 5-methoxy-U can be used in lieu of this. I think my overall takeaway here is designing your mRNA on the front end is extremely important for the expression and use downstream. Post-transcriptional modifications can be used, such as purification, but if you're able to design your mRNA properly on the front end, that's going to help you out significantly in the long run. All right, and I think that just about wraps us up here. So here we have our sales address, our sales email address, and our phone number. Feel free to reach out to us at any time if you have questions about the presentation, if you would like a quote, if you would like more information about any of our products, not only NTP, but also CleanCap, mRNA, custom mRNA. Feel free to reach out at any time. We'll be happy to get back to you and start that discussion. And then, lastly, I wanted to highlight some of these upcoming basic mRNA basics webinars. On April 13th, also at 9 a.m. Pacific time, we have capping strategies for mRNA manufacturing. We went into that a little bit today, but this one will be much more encompassing of the capping strategies. And then on May 19th, we have optimizing your mRNA coding sequence. So this will go into U depleting, codon optimization, and some of the strategies, again, that you can use on the front end to really maximize your mRNA results. Okay, I think that's it. We can get to some of your guys' questions here. Turn it over to Tina.
Great. Thank you, Corey. That was a great presentation. And thank you to everyone that's been sending in their questions so far. I see we've got quite a few questions here already in the chat box. Please feel free to send in more questions, even if we don't have the chance to get to them all today. We will do our best to follow up by email after the presentation. All right, so our first question that we've got here, are there any intellectual property constraints when ordering mRNA or CleanCap from TriLink?
Sure. So it is important to note that CleanCap technology is patented. We only require a one-time licensing fee at the time of commercialization of your product. So when using the product in a research grade setting or even in early phase settings, there is no licensing fee. When you want to take your products to a commercial setting, that's when we will require the licensing fee.
All right, thank you. So our next question here, in terms of production yield, do base modifications reduce the transcription efficiency of the IVT reaction?
So that's a good question. You know, some do and some do not. Some NTPs that have large or bulky side groups can exhibit lower yields. There is a chance that, you know, especially with some dye-labeled NTPs, such as cyanide-labeled NTPs, these are very large, bulky groups. They can inhibit the ribosome during translation, but mostly with the, you know, standard, you know, pseudo-U, these types of NTPs, we don't see a large yield hit. Your yields are probably going to be pretty comparable to the wild type counterparts here, unless you're using, you know, some larger or bulkier NTPs.
All right, thank you, Corey. Next question here. Does using modified NTPs with a U-depleted sequence increase the expression of mRNA above modification only or U-depletion only?
So yeah, so from that one slide, I can go back here because I think it's good showing. So, you know, when you combine the two, we do see here that in this case it does express, you see higher levels of expression when combining both a modification and the U depleted sequence. But this is not going to be, you know, this isn't a one all be all for every sequence, but I think it is a good place to start for most sequences. We recommend U depleting our sequences or your sequences for our catalog mRNA. We've U depleted all of the sequences as well as added on a 5-methoxy-U modification. So it really is a good place to start. If you're looking to test this out, my advice would be to try, you know, some head-to-head experiments or a screening study along those lines.
All right, thank you, Corey. So next question here on CleanCap. Is CleanCap available as a GMP version?
Yeah, it actually is. So it's been used in several clinical trials and with GMP grade material. It's actually been used in some of the COVID-19 work that's been going on. So absolutely, it's available at GMP grade.
All right, thank you. Our next question here on the topic of U depletion. What percentage of U depletion is ideal? Does it need to be complete or is there a medium somewhere?
Yeah, so there's definitely a happy medium, and this is going to be sequence dependent. I think, you know, for our sequences, we have these around 15% U content, 12 to 15%. And I think, you know, one strategy that I like to give out is trying to keep that uridine level below 20%. If it's possible to codon optimize and deplete the U level below 20%, you should be pretty good. But oftentimes, you know, the more is better. Some of our catalog mRNAs are U depleted to 12 to, like I said, 12 to 15% there.
All right, thank you, Corey. And we've got another CleanCap question here. Will CleanCap AG not work if you have a GG sequence?
If you have an AGG sequence, so AGG is actually the recognition site. So that will certainly work with that. That's the recognition site that we actually prefer to tell our customers. When we do start with AGG, it binds to AG and it's called CleanCap AG, but yes, AGG will certainly work.
So I believe they're asking about a GG sequence.
Oh, sorry, misheard you. No, so actually that's a good point. No, it will not work with a GG sequence, and you will need to actually make that shift to an AG sequence for it to have binding capabilities.
What about using something like CleanCap GG?
CleanCap GG will bind to a GG start, but you do see some of the same, same issues as ARCA, a bit lower capping efficiency, but it's good to note that this is still going to be a Cap1 structure.
All right, thank you, Corey. And I see we are running out of time here, so we've got time for one more question. So the question here, what type of assay can be used to measure capping efficiency?
Sure. So TriLink has a proprietary assay in-house that we use. It's our capping assay. Essentially, you use an enzyme to cut the 5' end off of the mRNA with part of the 5' UTR. Then you measure this via LCMS to test the relative ratio of capped mRNA to uncapped mRNA. Knowing the weight of capped mRNA, knowing what it should be, you're able to determine this ratio and determine that it's, for CleanCap, we usually see 95 to 99%, but we can match that up to what we expect with the fully capped mRNA.
All right, thank you, Corey. And again, thank you so much to all of you in the audience for your participation in today's webinar. Please join us next month for the next webinar in our series. Thank you and have a great day.